All right, so recognizing our time constraints at this point, I'll be going a little bit quickly through here. First, I want to thank the committee for inviting us to be part of this discussion. I think there's a lot of details about how compounding occurs in a community practice or even an institutional practice that are very important to understand as we look to make any kind of decisions on the clinical utilization of these types of therapies. Gus gave a quick background on who we are as PCCA and our role in the marketplace. Quickly talking about the historical role of compounded pain creams in the marketplace. This all started in the 1990s. This is not a new invention. This didn't start with the insurance components that we've all heard about in recent years. And it started because of peer-reviewed published literature, specifically looking at the clinical utilization of anti-inflammatories in PLOs, pluronic lecithin organogels, and the clinical efficacy that was yielded from a transdermal delivery system that could be created in a pharmacy environment. So in the 1990s, that started to be utilized, and in the early 2000s, companies such as ours sought ways to improve upon that delivery vehicle. While PLOs had a degree of efficacy, there were some components of it which were less than desirable. The tactile sensation on it was pretty sticky, so patients didn't necessarily like that. It had a little bit of a grit to it. It wasn't a nice smooth preparation. So is that another area that we could improve upon? While at the same time maintaining or improving upon the actual delivery profile of both hydrophobic and hydrophilic molecules through the skin. So if we're going to look at the delivery of drugs through the skin, how do we validate that? How do we improve the chemical and physical stability of those formulations, the tactile properties? How do we validate that all of this stuff is actually happening, that the API are transmitting intact through human skin, and that the formulas are stable, that they're not creating incompatibilities with each other, that they're not precipitating out, that they're solubilizing, so on and so forth. When we look at the delivery vehicle of a formulation, it's critical to understand that the same active pharmaceutical ingredient can have transdermal or topical characteristics depending on the vehicle that it's in. And it's not just the solvent. So here you see 3 different FDA-approved drug products that contain diclofenac sodium. It's the same salt form of the same NSAID. And the top 2 are transdermal vehicles. One of them is a 1% gel, the other one is a 1.5% solution. They both use a form of alcohol in their, as a solvent system, and they're both for transdermal pain indications. The third one is, again, diclofenac sodium. At a higher concentration, it uses a different type of alcohol, but the formulation as a whole, it's not just the solvent component of it, it's the rest of the components of the delivery system are different, and this one is for actinic keratosis. This is not a transdermal pain vehicle or formulation. This is FDA approved for actinic keratosis. So your indication is very different with the same API, still using an alcohol-based solvent system, but the other components of your delivery system change the function of this formulation. That type of difference exists in compounding as well. So we have to be very intentional and knowledgeable about the type of systems that we are creating when we put together a formulation and the purpose for which that is built and the patient that we are treating. I'll talk briefly about the journal article that was published earlier this year in the Annals of Internal Medicine that had to do with the comparison between a placebo cream and compounded pain creams. This committee discussed this article briefly in the previous meeting in March, and I have also provided a summary to the committee. Hopefully you've had time to review that summary. There's some good information here. It does talk about differences in the outcomes of patient groups that were receiving different formulations versus placebo. They talked about a variety of different types of formulations. However, there were some systemic flaws in the methodology utilized in this journal article. For example, the formulations were chosen based off of a general diagnosis. If you had neuropathic pain, you were receiving a specific formulation. If you had nociceptive pain, you had a different formulation. And the amount of medication that was applied to each individual patient was not tracked nor measured. They were told to use a clicker type of dispensing device, similar to, I assume, what we've been talking about here in this panel. Yet they did not measure the number of clicks that an individual patient was to utilize. Therefore, we don't have a measure of the number of milligrams of these doses that each patient was exposed to. So if there was an adverse event, at what milligram strength of API were they experiencing that event? None of that was tracked. In addition, the way that the patients were instructed to utilize the medication was based on the size of the painful area. And I know that we're going to have a physician panel after lunch this afternoon, but when we treat pain, we're primarily focused on the intensity of the pain. Even your visual analog score, which is the most basic scientifically valid methodology of tracking pain consistently, utilizes the intensity score. They never asked patients about intensity when they either instructed them on how much medication to utilize, nor when they did their survey follow-ups on patients. Another component of this was a very low compliance rate, which the authors do acknowledge in this study. So while this is good data, it certainly has a number of limitations in it. And so when we would ideally design a clinical trial to look at the clinical utilization of compounded pain therapies, we must understand how we're treating these things. And I know that when we're doing clinical trials, we want to minimize as many variables as possible. And this gets back to Dr. Carr's presentation from this morning of what we do in the real world versus how we conduct trials for clinical trial settings and for reporting, and the number of variables and the amount of patient interaction, the titration of doses. Those are things that we do in the real world that many clinical trials avoid for confounding variable purposes. So looking at the real world, and when we're talking about the utilization of compounded pain creams, there are a lot of FDA-approved products for the treatment of pain. And not all of them are topical. There's various modalities that physicians and pharmacists have at their disposal to help our patients feel better. Pain is very subjective. The intensity of pain, how pain is reported, is largely subjective. And the FDA-approved products do take care of the vast majority of our patients. As Mr. Vallava, Dr. Vallava mentioned earlier, this is something that is typically a last resort for many patients. These are not typically naive patients to various types of modalities of medications. So it's that small percentage of the population whose needs are not met by the FDA-approved products who then seek alternative solutions to their issues. There's a lot of different reasons that comes into play. The two most dominant ones have to do with the route of administration and effective dosing to control the pain while minimizing an adverse event profile. I do have some videos here that talk about how the compounds are prepared, the type of equipment that is utilized. We don't have a lot of time. We're already kind of over time, so we're not going to show the videos. I've submitted those. You have access to those, certainly. So you've got those videos at your disposal. You have access to those. Compounding is not manufacturing, right? So a lot of those discussions about the level of data, we have Franz diffusion studies on a number of different formulations. We've got full stability indicating assays and methodologies to validate the physicochemical stability of the formulations. And these are things that are looking at the integrity of the process and ultimately how we can ensure consistency for the formulations that are being dispensed to patients. And when we develop those types of formulas, we are very transparent with all of the methods that are utilized, the specific equipment that was used, both in the measuring, the mixing, the packaging, the storage conditions, everything. We talk about the specific settings of that equipment. And we have explicit notes on all of these types of formulations that say if you make any alterations to this, if you're not using the right equipment, if you're not using the right chemicals, if you're substituting specific components, then the data that you have here is no longer applicable. So we're very intentional in how we're providing this type of information to the pharmacist so that the scientific approach is maintained from start to finish. There's also third-party testing. So the pharmacists in the field, three of whom are on this panel, they do utilize third-party FDA-registered analytical laboratories to validate that their formulas are maintaining the characteristics that they expect when they followed a recipe or guidance from an outside reference. So with that, I will stop my comments and leave time for a committee discussion.